Welcome, everyone, for another dose of science. And we have an interesting one for you here today. Joining me, a little bit of a different crew. Emily is still on mat leave, babying. Jonathan is on vacation. And sometimes we let our people take vacation. And we have Ada back with us, who, of course, is a regular. And we are joined today by Gaia Remerowski. Gaia is a science communicator with us here at the office, but she's actually in St. Louis. So she helps us remotely. But Gaia was my first ever hiree here at McGill. And that goes back, I think, 21 years. And, you know, they've said that you can kind of judge people by the success of the people that they have trained. And so I do claim success here because Gaia has done very well. Ada has done extremely well. All my others have done well as well. All right, Gaia, welcome. So before we do anything more, give us a little bit of story of your evolution after you left McGill and how you arrived at where you are now.
Sure, yeah. So it was a little circuitous of a route, but I started off going to graduate school in Boston at Boston University. I did a degree in science journalism, which was very appropriate. Joe had prepared me well for that. And then I went on to work in public television. I worked for the science show here in the US called Nova. On PBS, and I did story research there. And so I found ideas for stories that we could tell on the show and help fact check those stories. And then my lovely husband, whom I met in Boston, moved us to St. Louis for his job. He was a scientist working at Monsanto at the time, which is now has been acquired by Bayer Crop Sciences. And so that brought us to St. Louis, where I currently live and work. And I started out at Washington University School of Medicine here in St. Louis. And I was doing communications for a group called the Genome Institute, which, interestingly enough, helped sequence the human genome way back when. And so I was helping them kind of get more awareness about what they were doing and telling the public about the kind of slightly more complicated science that, you know, they were doing. It's tough to explain genomics to the lay public, but it was a challenge I really enjoyed. Then I moved on. Since then, I moved to the main office of public affairs at the medical school, where I generally communicate scientific and medical topics for the entire School of Medicine now. And that's what I've been doing for a while. And so that's sort of my current situation right now. And then I reconnected with Joe, and I'm happy to be helping out, writing some stories for the Office for Science and Society as well.
There we go. Full circle. Right?
Yep.
Full circle. All right. So communicate. Tell us the story that you're going to discuss today.
Sure. So I thought I'd talk about a study that came out last week that suggests the SARS-CoV-2 virus, which is responsible for COVID-19, might have arrived in the US earlier than we actually thought. So the first confirmed case of SARS-CoV-2 was, I think it was identified on January 21st of 2020. And it was a patient in Washington state who had recently returned from Wuhan, China. But in this study, what the researchers did was they took frozen samples from patients who had donated blood between January and March of 2020. And they looked at those blood samples and they searched for antibodies to the SARS-CoV-2 virus. So just a reminder, antibodies are proteins your body makes to fight off infections caused by things like viruses and bacteria. And so of the more than 24,000 samples that they tested, they found 9 people who showed antibodies to the SARS-CoV-2 virus. And these 9 people, interestingly enough, were from 5 different states, none of which were Washington state. And so the 5 states were Illinois, Wisconsin, Pennsylvania, Mississippi, and Massachusetts. And all of the samples were actually taken weeks before any of those states had identified cases in their state. So, for example, the Massachusetts blood sample was taken on January 8th, but the first case reported in Massachusetts wasn't until February 1st. And so it actually takes about 2 weeks for antibodies to develop to the SARS-CoV-2 virus. So many of these people probably actually developed the infection more likely closer to the end of December of 2019. And another study that was published before this one actually found that some people did have antibodies to the virus as early as December 13th of 2019. And so if you recall, early on in the pandemic, it was really hard to get a test for the virus, partly because there weren't that many tests that were available and approved yet. There was supply issues for the materials used to make the tests. Some of the tests were inaccurate, and there was also a policy in place that said that you could only get tested. This is certainly in the US. I'm not sure about Canada, but in the US, there's a policy in place that said you couldn't even get a test unless you had traveled to one of the countries that had a very high infection rate, like China, or that you had come into contact with someone who had traveled to one of those countries. And so all of these kind of setbacks with testing and the policies in place at the time, this study is basically suggesting that those might have hidden the fact that the virus was here earlier than we actually realized. And an interesting side note about the study, the samples came from a different study that's actually ongoing. It's called the All of Us Research Project, and it's a study that started in May of 2018. And it's funded by the National Institutes of Health. And the goal of it is to collect data from 1 million diverse Americans across the country. And what they're doing is they're taking blood samples from these people, they're getting their health records, they're surveying them to learn about their diet, their exercise, environmental exposure. Basically, they just want to know as much as they can about health and well-being across the US. And another interesting point is they really wanted to make sure that the study was representative of race and ethnicity across the United States. And what's interesting is that turns out to be a really good proxy for studies like this one that just came out, looking for the antibodies to the coronavirus, because what they found in this recent study is that of the 9 people who showed antibodies to the virus, 5 of them came from samples of people who identify as Black or African American, and 2 of the 9 samples came from people who identify as Latino, Hispanic, or Spanish. And that's actually really interesting because that's very indicative of the course of the pandemic and how it has disproportionately affected those races and ethnicities. So that was also an interesting point. A couple of limitations to the study. There are 4 other known coronaviruses that infect people in the US and cause common cold symptoms. So it's possible that what the researchers were detecting were antibodies to those other coronaviruses. And that would be what's called a false positive. But to help control for this, the researchers actually took blood samples from early 2019, long before the pandemic started, and tested those for antibodies as well, and did not find any positives. So you would expect, if these other coronaviruses are so common, you would have at least gotten some positives. Another thing they did to cut down on testing errors is they made sure to test the samples with 2 different platforms, just to make sure that there was nothing wrong with the test itself. And that's actually something that the Centers for Disease Control and Prevention recommends people do when they're testing for antibodies.
Obviously, there's nothing here that contradicts this coming from China. I mean, that is well established. The only question is when it came. And it looks like it came earlier than everyone thought, which just underlines the fact that the president knew about what was happening much earlier than what he said he knew, right?
Right.
Yeah.
Yep. Yeah. And then another, drawback to the study too is the samples didn't, the researchers don't know if the samples that tested positive if those people had traveled or if this was community acquired, if it had spread amongst the community. So they are going to go back and try to determine the status of, who had traveled and who hadn't among those 9 positives. And then one last point is just, they did test over 24,000 samples and only 9 of them did turn up positive. So even if the virus was here a lot earlier than we thought, it's clear that it wasn't widespread yet. But I think what it does show us is that it's really important to have those case reporting and testing policies in place early on in pandemics like this so that we can better understand the spread of the disease.
All right, let's go to Ada, our COVID expert, because she's the only one who's actually had this.
This a personal expert.
Yeah, personal expert.
But that's really interesting. So I got my handy dandy calculator, and that's less than 0.03%. Yeah. So it's really such a minuscule number, but it does seem to make sense with the anecdotal reports we've seen. Even reports in case studies and on Twitter, kind of just word of mouth. A lot of people are talking about getting a really, really vicious cold or flu type thing in really early 2020 before we even knew that there was a pandemic about to happen. So at least anecdotally, it definitely lines up.
Well, and the other interesting thing is that they did look at health records of these people who tested positive, and only one of them showed that they had gone and received medical care for symptoms. So that's not to say the others didn't, but it didn't show up in their health records.
That kind of makes sense to me, though, because if you expect so many cases of COVID are able to be asymptomatic or mild symptoms. I mean, even mine was pretty mild in the end. If it's only 0.03%, there's a pretty good chance all of those could be asymptomatic or very mild. And I mean, before the pandemic, not a lot of us really freaked out about every cough and cold anymore. I used to get sick like once a month, and that was so normal, and that feels so weird now.
All right, let's rain on the parade here. Ada.
Oh, no, you can't rain on my parade. I'm good. I'm prepared. This is bad luck, I think. It's a good thing I don't believe in those things.
Right.
So I've got today kind of an interesting little tidbit. I really like to myth bust, but it's getting a little bit too easy almost to kind of myth bust things, pseudoscientific claims. Homeopathy, we've talked about it a lot. We've really kind of done to death a lot of the pseudosciences, and there's more ones being made up every day. But my personal favorite myth busting are the things that we don't even know to question. So the things that it's not even like you could say, well, maybe homeopathy works. I don't know. It's something so ingrained in us that we don't even think that it could possibly be wrong. And so pretty much everybody, including quite a lot of meteorologists, have a really fundamental misunderstanding about what probability of precipitation even means. So I think a lot of people, me included, we think that basically if there's a, say, 40% chance of rain in Montreal, which is about what there is today, that means that in Montreal, if I stay outside all day long, there's a 40% chance that I will get rained on. But that's not actually quite right. It turns out, despite what you think, a lot of people have this idea that meteorologists are just kind of wrong. Like yesterday, there was a severe thunderstorm warning all day for all of Montreal and nothing. I left my laundry up on the line all day and it actually didn't even get wet. So I think a lot of people often think meteorologists are just kind of mistaken, or that predicting the weather is more of an art than a science. But it turns out most people actually just have a bit of a misunderstanding about what a PoP or probability of precipitation, because it's not just rain, it could be snow or sleet or other forms of precipitation, what it actually means. So there's a really simple formula, but it's kind of a little bit to wrap your head around it. So we'll go through it together. So the probability of precipitation, the PoP, is the percent coverage times the percent confidence. So as an example, if we were 100% beyond certain that it will rain in Montreal, but only on 50% of Montreal, then the PoP for Montreal for that day is 50%. But depending where you live, if you live in the part that's predicted to be rained on, your PoP is, in the sense of how we think about it, as in the chance that I will get rained on, is actually 100%. But if you live in the other side, your chance is actually much lower. So we don't think of this kind of area effect that comes into play with chances of I keep wanting to say precipitation. So another example, I live in the borough of St. Marie, and you can choose, and this is one reason why it's actually important too, you can choose what area for a weather report that you look at. So most of us, I think, probably look at Montreal or St. Louis for you, Gaia. But I actually choose to look right at my borough because you'll get a more specific report the more you zoom in. You can do the opposite too. You could look at the report for all of Quebec, but that's not going to be very specific to you in Montreal. So if we're 20% certain that it'll rain on most of St. Marie, so we're not super confident that it'll rain, but if it does rain, it'll be on like the entire borough of St. Marie, the POP would only be 16%. But that still means there's a 20% chance, because we are 20% sure, that it will rain on most of the borough. So you could see this POP of 16% and there could be no rain, and you'd be like, yeah, okay, that's what I expected. Or you could get totally downpoured on and be like, what the heck? They said it was a 16% POP. But that's kind of just the trick of how this POP works. And that's why I actually prefer to look at weather radars, because it's a lot easier to just look, especially if you're doing something in the next couple hours. You can just look at the trends and the way that the weather's moving and make your own judgments. So for today, we have a 30% POP for Tuesday evening. That could be 100% confidence that 30% of Montreal will receive rain, or it could be 50% confidence that 60% of Montreal will receive rain, or it could be pretty much any other number of combinations, because that's how math works. So unfortunately, it turns out, at least to me, POP is not really that useful in terms of knowing if you'll actually get rained on. So my advice is to either check the weather radar or just carry the umbrella with you all the time. They make them pretty small now.
I'm usually 100% accurate because I do it by looking out the window.
You stick your hand outside.
And I can. All right, let me share my my screen now, and okay, there we go. And I want to tell you a little personal story, because way back some years ago, when I was, guess about 6 years old, I had my tonsils out. And this was back in Hungary. This was in the 1950s. And I was put to sleep with chloroform. It was a pretty primitive technique back then. They took the chloroform, they put it on a gauze, put it over my mouth, asked me to count backwards from 3, and I probably got to 2. Now, luckily, this was before chloroform was carcinogenic, so I don't think it did me any harm. But that's how it was done in those days, and my tonsils were whipped out in a minute and a half, which was a record at that time. Well, chloroform had been invented as an anesthetic in the middle 1800s by James Simpson in Scotland, the Scottish physician. And in those days, they just tried all kinds of things to see whether or not they would be able to somehow put themselves to sleep to take away pain. Today, of course, chloroform is no longer used because it is pretty toxic and we have much better anesthetics. It doesn't mean that it has no uses. Chloroform is used today, for example, to form tetrafluoroethylene, which is the monomer that is used to make Teflon. And as you know, there's controversy here about these fluorinated compounds, which we've talked about before. Undoubtedly, we will talk about it again. Anyway, that's another story for another time. But after I had my tonsils out, I was given lots of cream to eat because that's how it was done in those days. You know, if you had pain in the throat after tonsillectomy, they would give you the ice cream because it would momentarily freeze it. But I had an aunt in Canada who learned about my little surgery here and sent us a sample of aspargum, which already was pretty popular here, and it was the treatment for after tonsillectomy. So let me just tell you a little bit of a background here about aspargum. obviously it was based upon aspirin. And the discovery of that drug really goes back to the 18th century and the Reverend Edward Stone in England, who found that the bark of the willow tree, when ground up and extracted, had something in it that was amenable to relieve pain. But it was quite a process before that willow bark got turned into aspirin. And that was thanks to Felix Hoffmann, a chemist working at the Bayer company, whose father had a history of rheumatic pain. And in those days, they would give willow bark for that pain. But the trouble with the willow bark was that it contained salicin, and salicin was very irritating to the stomach, although it did relieve pain. So Hoffmann decided that he was going to do something about this. He tried to somehow improve on salicin, which he managed to do, and he made a derivative of it called acetyl salicylic acid. And this became aspirin. The A from the Latin for from, and spirin from the Latin for willow. So although aspirin was never found in the willow tree, it was used to, at least a chemical in the willow tree, salicin, was used to formulate aspirin at first. And then other techniques were developed. Interesting enough, Felix Hoffmann also synthesized heroin. And in those days, the Bayer company sold aspirin and heroin, on the same bill, interestingly enough.
But was there heroin gum?
Yeah, the heroin gum. No, there's no heroin gum, but there's white willow bark that you can buy in the health food store. Now, this is one of the biggest absurdities. Why? Because the reason for development of aspirin was that white willow bark was not very good because it was extremely irritating to the stomach. But today, this is perceived as sort of a natural remedy. Well, in this particular case, the synthetic aspirin is a lot better than the natural white willow bark. Anyway, back to my tonsil and aspirin gum story. An American physician in California, Dr. Lawrence Craven, used to give aspirin gum to his patients after they had had a tonsillectomy. And he noticed that this increased the chance of their bleeding. Now, most people would have said, oh my goodness, that's not good. I better stop giving aspirin to these patients. But he saw that there was a golden nugget in there, because he recognized that there must be something in aspirin that reduces the chance that blood will clot. And he was very familiar with the fact that heart attacks can be caused by blood clots. So he started to give aspirin to his cardiac patients in tablets, and he saw that it reduced the risk of heart attacks. Today, of course, this is well known. And there's a strong link between blood clot formation and heart attacks. And we know what the link is. Dr. John Vane in 1971, who eventually got a Nobel Prize for this, figured out that aspirin inhibited an enzyme, and the inhibition of that enzyme was responsible for the effect on the blood clotting. And today it is very common for patients who have already had a heart attack to reduce the risk of a second heart attack to give them a small dose, 81 milligrams of aspirin. So that's my story. And I first learned about aspirin gum, of course, through that tonsillectomy. Not at that time. I had to reflect upon it later. But I think it's sort of a interesting story because of the serendipity involved. Craven was not looking for a way to reduce the risk of heart attacks. He happened on it accidentally. I'll give you another story. And for this, we'll go to Johns Hopkins University in Baltimore. And this was the discovery of saccharine. Dr. Ira Remsen was a well-known professor at Johns Hopkins at that time, and had a postdoctoral fellow, Konstantin Thalberg, working under him, who was trying to carry out a synthesis of a molecule called toluene sulfonamide. And I guess he didn't have very good hygiene habits, because he ate his lunch in the laboratory. I guess people did that in those days. And when he picked up his sandwich and bit into it, he found that it tasted sweet. Now, of course, many people would not have paid any attention to this, but he backtracked, and he recognized that it must have been the chemical that he was working with that caused this, and that was the birth of saccharin. And something that many people don't know, and Gaia, this will be of interest to you, is that the first substance that Monsanto ever sold was saccharin. In fact, the Monsanto chemical company was built on the back of saccharin. Monsanto was the family name of the wife of the gentleman who started the company, and their first product was saccharin. And in a very similar way, in 1965, Jim Schlatter, who was working for the Searle Company, also because of questionable hygienic habits in the laboratory, he was doing some research on gastric ulcers. And he was a smoker. And he would be working in the laboratory and smoke, and he found that the cigarette had a sweet taste. And this eventually turned out to be aspartame, the chemical that is 180 times sweeter than sugar. So sometimes, accidental discoveries can become very, very big discoveries, as in these instances. So you kind of never know the road that science will take. And there are many, many other examples, of course, in the history of science based on accidental discoveries. And Ada is making discoveries every day now. They're not accidental, right? They're purposeful.
They're purposeful, but there are lots of accidents along the way. Ironically, I also use a lot of chloroform. So that has found at least a modern use in laboratories, to be sure.
Yeah.
I think my favorite serendipitous discovery from science is the LSD story, where the poor, I forget his name, I'm not good with names, but the poor researcher, he tasted the drug, started tripping out, and got on his bike and tried to bike home. And that's why we still celebrate Bicycle Day in some places around the world.
Yeah, the other memory I have of chloroform is that if you get it in between your fingers here, it burns like crazy.
That's why I wear gloves.
Yes, well, in my days, we didn't wear gloves, and we didn't have fume hoods. We ran chromatography columns with benzene and chloroform out on the bench, so certainly things have changed. But I tell you, one thing that hasn't changed is the dissemination of nonsense. And the reason I bring that up is because last week, of course, we saw this horrible picture. Some of us saw it live, of the Danish soccer player who collapsed on the field. And he had to be resuscitated. It was actually a cardiac arrest. But the troublesome part of this story is that within minutes of this happening, a message started to scoot around the internet that he had collapsed because he had gotten the COVID vaccine. There's absolutely no truth to this. First of all, the COVID vaccine, of course, doesn't cause that, but he did not even have the vaccine, and he had not had any COVID. But what is interesting is how such a story flames up so quickly, and all you need is one blogger to start this. It's really, and this is, at our office here, this is the bane of our existence, is how these stories get spread around. And, things like supposedly the chief executive of Moderna said that the vaccine changes DNA. He never said anything like that, yet this is all over the internet. It goes back to a TED talk that he gave a few years ago, where can make the body form all kinds of interesting substances. He never talked about DNA, but there it is. It's all over. So Gaia, I'm sure you see this all the time in the med school surroundings, right? Where the ridiculous nonsense just takes off.
Oh yeah, absolutely. And we're constantly having to respond on social media if we publish something, a story about a new drug or a new finding in animals or something like that, and people start asking, well, first of all, they're saying, well, why can't I just use my herbal remedy that we get that a lot. So I remember we published something on a possible new therapy for eczema, which is a very common skin disease. And apparently there's a huge community out there that are using this special herbal remedy for eczema, and the comments just started flooding in. Like, I don't need any drugs. I've got my natural way of solving it. And the tough thing with eczema, it's one of those things that comes and goes naturally, right? And so it's really easy to say, well, look, it helped me. It's like, well, maybe doing nothing could have helped you as well, right? So that's really difficult too. We get a lot of that, but absolutely, we have lots of comments, and often it's on social media. And those are the ones that, we feel like we have to address.
The misinformation works two ways. You can have misinformation about something that really works, and the misinformation is that it doesn't work, such as with the vaccines. And then you have the other part where you have no evidence that something works, and yet the bloggers, of course, are claimed that this is the newest miracle. I just had a question about red seaweed. Whether or not we should be eating this every day, because a study came out, which was an interesting study. It looked at the rate of colon cancer in Japan, which is actually amazingly low. And the Japanese eat a lot of seaweed. Of course, there's the association, but obviously association is not cause and effect, but it means that it's worthwhile looking into. And there was a research paper that came out that looked at the composition of red seaweed and found that there is actually sugar in there called agarose, which is a prebiotic. So it increases the food supply for the, quote, good bacteria in the gut. And you can make a case for, potential benefits. But that doesn't mean that we should start guzzling seaweed. There is something there that needs further exploration, but people are so ready to jump on the miracle bandwagon. And anytime there's any kind of study that suggests some tiny benefit, they will take off with it.
Well, isn't it true that the Japanese also have an enzyme that allows them to digest seaweed that we do not have as well? And so that's another thought.
Sure. I mean,
I don't know about you, but whenever I eat sushi, I sometimes, especially if it has a lot of seaweed, sometimes I don't feel so good after. Because I think, there's something to that where you can't, like, I don't know that in North America we can digest it.
Do many other things differently than we do. So you can't focus in on just one food.
Yeah.
Anyway, I never liked seaweed. It probably is because I have a fish allergy, and it just reminds me of the ocean taste, and that bothers me. Ada, you eat sushi?
I do. I love sushi, actually. And I'm a vegetarian, so it's actually a pretty important source of seaweed, can be a pretty important source of nutrients for people eating a plant-based diet.
Oh, there's certainly nothing wrong with eating.
I kind of hate it, though. I don't like how it tastes.
All right. Well, talking about food, I have one other story here. So let me just share my screen once more. Let me introduce you to Benjamin Thompson, who was also known as Count Rumford. Rumford was a town in New Hampshire at that time, way back in 1700s. And when he was made a count in Bavaria, he was given the choice of taking on a name, and he just liked the town where he had his beginnings, and he chose Count Rumford. Anyway, he was born in the U.S. He was born actually in Woburn, Massachusetts. The house is there. And I like his history because it's very interesting, and you'll see why. During the American Revolution, he did not side with the revolutionaries. He was a loyalist. He sided with the King of England, which obviously was not a very popular thing to do at that time in the US. So he had to flee after his house was attacked by a mob, and he fled behind the British lines. They welcomed him because he had a scientific background, and he was doing research on gunpowder. So they wanted ways of making better gunpowder, so they welcomed him. But after, of course, the British lost the Revolutionary War, he had to leave America. He went to Britain, where he became the chief scientist to King George III. And he did some interesting things. For example, he was very big on coffee, and he thought that coffee was great for headaches, and there is actually some evidence for this. And amazingly, he developed the coffee percolator. That was his invention. He also
Thank
had
you.
a way.
Thank you very much.
Yes, percolators are great. And he also invented a fireplace, or at least the configuration of a fireplace, so that smoke would not come into the room, because in those days it was very common for the residence to be filled with smoke. But in this cartoon here by the famed British cartoonist James Gilray, shown here standing, next to the Rumford stove, as it was called. Anyway, he had a bit of a falling out with King George because it seems that he sold some naval secrets to the French. The king didn't like that. And he banished him from England to Bavaria as a diplomat. He wasn't really punished because he agreed to work as a spy for the British. But once he got to Bavaria, he forgot about the Brits, and he became an advisor to the Duke of Bavaria, and eventually he was made a count, and that's when he chose the name Rumford. And he was given the task of taking the poor off of the street. So he developed these workhouses where the poor would work for low wages, and this was very good for the state because they got cheap labor and the poor at least got some money, but then he had to find a way to feed them in these workhouses. And he developed what came to be known as the Rumford soup, and there are still many recipes for this. It was made with barley and peas, and in those days, this was thought to be nutritious, and actually it isn't bad. It was cheap, it had potatoes in it as well, and this is what he fed people in his workhouses. And then he developed a machine to dry potatoes. And he thought, maybe this could also be used to cook meat. So one day, he took a piece of mutton, which is leg of sheep, and he put it in his potato dryer. And he heated it up, fire underneath, and he checked it about 3 hours later, and it turned out it wasn't done. So he said, I don't need this. He left it to the maids who worked in the kitchen, and he said, just turn off the fire, and he left. Well, they thought that after turning off the fire, they might as well leave that meat in that contraption because it was as good a place as any other. And then they came back in the morning and they were surprised because what they found that the meat had cooked perfectly. It was succulent because, of course, this iron contraption retained some heat, even though they had extinguished the fire underneath it. So what you had here was long-term slow temperature cooking. And Rumford became excited about this because he had long had the idea that, well, it was very common practice that whenever you boiled any vegetables, potatoes, anything, you had to do this at a vigorous boil. And that never made sense to him that the ideal temperature for cooking would be the boiling point of water. So he began to do a number of experiments whereby he would cook food at a simmer or even at a lower temperature, and found that it became more succulent and more tender, but of course it had a longer cooking time, but he also had to use less fuel, which was important in those days. Well, believe it or not, this was the birth of a modern cooking technique, which is used by the most exalted chefs today, and that is known as sous vide. Sous vide is cooking at very low temperature, and it is usually done with putting the meat or the vegetables into a plastic bag and immerse it in water. And the contraption that you see here keeps the water temperature exactly where it is set. So very often it is cooked way below the boiling point of water. There's also a rotor in there that circulates the water. You get very even heating. And the idea is that you get the best possible taste for the meat when you cook it like this, and you're also saving some energy. There's one problem, that you don't get the browning. The browning is what we call the Maillard reaction. It's when amino acids react with sugars in the food, and you get the crust like on bread or the grill lines on meat. That also adds some flavor. So the cooks now will do the sous vide cooking, and after that sear the meat very quickly for a very short time. And that apparently gives you the ultimate flavor. So there's the story of Benjamin Thompson. And again, it was an accidental discovery. of having left that mutton in that potato dryer overnight. But finally, another note about Benjamin Thompson is that together with Sir Joseph Banks, he founded the Royal Institute of Great Britain, which is dedicated to disseminating good scientific information to the public. And the first lecturer that they hired was Humphrey Davy. And again, depicted here in this classic cartoon by James Gilray, where you see the impish Humphrey Davy, one of the greatest British scientists of all time, having just cooked up some nitrous oxide, which the unfortunate volunteer from the audience is inhaling, and it is making a rather dramatic exit from his rear. But people flocked to these Friday night demonstrations at the Royal Institute to learn all about science, thanks to Benjamin Thompson, Count Rumford, and a statue of him is right there in front of the library in Woburn, Massachusetts, where he was born. And I just found out that there is yet another side to Thompson, and that is he was a real ladies' man, as is depicted in this book, which I have just ordered. So maybe sometime I will be able to tell you about his exploits, not only in the kitchen, but in the bedroom as well. All right, Ada, Gaia, have you ever cooked sous vide?
I have not. No.
I don't eat meat, and it doesn't. I know you could do it with vegetables, but I never see someone saying this just like changed the way a potato is prepared. You know, it's not quite the same level of difference as it is for steak, I think.
I've never tried it, but I'm willing to give it a shot, just obviously for the scientific interest here.
Well, and I assume that there's no food safety issue, right? Because if you're searing it, you would kill any bacteria, right?
Right, you would kill any bacteria. And also, I mean, you're cooking it long enough, so it does get pasteurized. The one issue that does come up is whether or not there's any chemical being leached out from the plastic when you're doing this, which is, of course, is a reasonable question to ask. Because, you know, when we're looking at concentrations of parts per million, it's possible that but these bags are made of polyethylene, and polyethylene does not leach out any kind of estrogenic compounds. That might be an issue with PVC, polyvinyl chloride, or with polycarbonate, but not with polyethylene. So I think it's safe enough and interesting enough to try once, although I doubt that it would make a huge difference in the flavor of food, but there are restaurants where the claim is that they serve really special food because they cook it sous vide.
Well, I have had a sous vide cooked egg, and it was really perfectly cooked. It was like a poached egg, but I don't know, it didn't have the kind of liquidy, watery part that you have sometimes.
I can tell you that there are dozens and dozens of sous vide videos about how to cook an egg It's
really hard to poach eggs.
It is. Yeah. You want to have just the right texture. Absolutely.
I have this little silicone like cup thing that I like float in the pot of water like a boat and put the egg in there to poach. So isn't that essentially sous vide? Maybe I have had sous vide.
Yeah. If you're poaching it at the boiling point of water, then it's not sous vide.
Yeah.
So it would have to be just warm water for a long time.
Slow cooking isn't quite the same thing, but I do really love my slow cooker.
Yeah, but for the poaching eggs, isn't it true that the hardness of the water will affect it too? The poaching?
In theory, it would, because you have calcium and magnesium in there, which would alter the way that the proteins are denatured. Egg cookery is a whole world in and of itself. I just like the hard-boiled egg. I leave it in there for 12 minutes, and it's good.
I do them in my electric kettle. Just pop them right in the kettle while I make tea.
We have run out of time here. And Ada, a final word.
Pigeons. That's the word. That's it. That's just the word pigeons. That's all you get. No, just kidding. So pigeons, one reason why they're so successful in urban areas, this is a 2-for-1 fact because I love pigeons. So one reason they're so successful in urban areas is that they're able to process like toxins and heavy metals that they may get them through the air or through their food because they're eating kind of polluted water and food from a cityscape. They're able to sequester these toxins and heavy metals into the tips of their wings. So studies have taken pigeon wings and feathers from them and tested them for heavy metal concentrations, and it's massive. So it's actually a defense mechanism. And the other just interesting point of how pigeons wound up in cities at all is actually that pigeons aren't wild animals. They're more properly feral animals, because a wild animal is an animal that was never domesticated and remains wild. So things like wolves, foxes, polar bears, they've never been domesticated and they still are not domesticated. Whereas pigeons were domesticated. We brought them over here and then we said, what the heck? Who put all these pigeons around? And now we hate them, even though we brought them here. So they're technically feral, just like you would call a dog or a cat, which were domestic animals that run free around the city. They're feral dogs and cats. So pigeons, not wild, yes, feral. And you should be nice to them because they used to be our pets.
And the eternal question, how do you clean pigeon poop off your car? And
I recommend not having a car.
Windex works very well on pigeon poop. And you want the final word on pigeons? I'll give you guys something interesting to read about. Read about Tesla, Nikola Tesla, and pigeons, because he communicated with pigeons. All right, that is it. We've run out of time. We will do this again. Thanks, Ada. Thanks, Gaia. And thanks all of you for watching. And thanks to Stuart for putting it all together. See you next time.